right. Let me see if I can get things going, and let me go to Dr. Benedict. And in your presentation, you talked about the need for containment, and you talked about how a current system that has, for example, a BSL-2 lab, and one of the problems that you mentioned has to do with flying insects, for example. Do you imagine some sort of a variation is needed in terms of the current way of classifying labs, or is this the sort of work that you would recommend being done only in BSL level 3 labs, or what's your thinking about that on a practical level?
All right. So I do think a lot of the burden is going to fall on institutional biosafety committees because there is no, as far as I'm aware, no clear guidance for this kind of containment. And I expect going forward we'll borrow less from biosafety guidelines than we borrow from, say, arthropod quarantine standards. And so I think that it's very understandable in the early days that the biosafety, the microbiological standards were applied to insects, but as I said in my talk, that they clearly have limitations. And similarly, working with mosquitoes under a BL3 compliance often is necessary. Let's say if you're working with certain pathogens and so on. But for conducting evaluations such as large population studies and things like this, operating at BL3 per se would be very, very difficult, I believe. So I think the standards that should be applied should maybe shift to a different model of containment, which would be more like what is used when, say, an exotic biocontrol arthropod is being considered for release, and how is it contained under those conditions? Because the problem is not a microbiological one. The problem is a species, an arthropod escaping into the environment. And so we need to think differently. And I'll give you a good example, and I think a very practical one. So the arthropod containment guidelines recommended negative air pressure. And you see this often mentioned as a containment measure for mosquitoes. Our laboratory has negative air pressure. Well, that's all good and well, and particularly if the mosquito is contaminated with a human pathogen or an animal pathogen. But it's been demonstrated that mosquitoes, for example, are stimulated to fly upwind, and this is their natural host-seeking behavior, looking for sugar meals, looking for humans and animals, and so on. So actually negative air pressure is much like to a mosquito, is once they sense that breeze, they're saying, there's a way out. And so so the point is that we need to be very thoughtful about considering the insect that we're talking about and what measures are really appropriate, and to what extent to the biosafety levels that are commonly applied for microorganisms, to what extent are they appropriate for insects? And so there's going to be a divergence between the current model of thinking in BL levels versus thinking in arthropod containment levels. And so I'm reluctant at this point in time to say, therefore we should do X, Y, and Z. But I do think we need to begin looking at a different model going forward, because we're not talking about preventing people from becoming infected with a pathogen or an animal. We're talking about the escape of an arthropod generally into the environment.
Right. Interesting. Thank you. Other questions from committee members?
Hi, this is Lisa Tannehill from the University of Maryland. Can you hear me?
We can, Lisa. Go ahead.
Great. I was just wondering, Dr. Benedict, if you could comment a little bit on the feasibility of being able to generate distinct visible marks in gene drive containing mosquitoes, for example, given that I'm guessing many of the transgenic strains may already have some visible markers. So how easy is it to do this so that one can discriminate a traditional transgenic containing mosquito from a gene drive containing mosquito?
Yeah, thanks, Lisa. That's a good question, and I cannot speak, let's say, for the Drosophila world. I think there are two cases, though. One is certainly in the mosquito world. Probably 95% of the strains that have been created in the past few years are marked with 3xp3ds red or 3xp3gfp, something like this, as the general transgenic marker. So the number of strains that use other markers is really pretty limited. Some of them have been demonstrated to work, but they just didn't get any uptake. And so the number of strains that exist is very, very small. So there are different, I think, different orbits in which labs might make this decision. A particular lab that works with a large number might try to get consensus inside the lab about what they will use, considering what they already are using. But certainly it'd be much more desirable to develop markers that were reflected a consensus opinion among the people who are working with a particular group of species that can mate with one another. So in another context, the reason that the mosquito people have focused on just these few markers is because we don't have such extensive studies of promoter utility for general transformation markers as, say, the Drosophila people have. So they're going to, we have a very small experience in comparison. The Drosophila people will have a large experience in addition to that diverse number of markers that they're probably already using. So I don't make that recommendation lightly, but given the value that such markers would have in assuring containment, I do think it's a very clear need that certainly the mosquito people, I believe, could reach consensus on, and would be very useful for containing the driving transgenes that are being created.
Great, thank you. Other questions? Let me go to Dr. Strauss. Steve, you talked about what you thought were real limitations of doing the sort of research that you described. And I may get this wrong, but I believe in more laboratory environments. And you described lots of work going on outdoors in the field. Is there an intermediate situation? In other words, large greenhouses, I mean really large greenhouses. I'm down here just north of the World is Biosphere 2, for example. And in some of the early discussions of CRISPR, there was pointed out the need possibly for national level, national scale supported facilities in which large experiments could be done, but in really protected environments. What do you think?
Yeah, I think. Am I mute? Can you hear me?
Yes. Yeah.
So my little mic mute button is on, so I wasn't sure. Anyway, I completely agree that we, particularly in the initial phases of gene drive work, say with a forest pest organism, that's the kind of environment you want, a very large, secure greenhouse, or a greenhouse in a place where that pest organism is not suitable for survival out of doors. Those are the places to start the work, and we do need those kinds of facilities. And on a case-by-case basis, whether we need some national facility for all sorts of work like that, or whether that can be done more locally, will depend on the risk considerations and all the stuff that you guys are talking about and how strict the containment needs to be. I can imagine doing work on, say, an invasive weed in Oregon in a standard greenhouse, given that the risk of some escape in terms of damage to the environment is very, very small compared to what what is going on currently. So, for example, for Scotch broom would be one type of example where I don't think anybody's talked about a significant benefit would be harming. So I could imagine a much less stringent kind of a containment facility for initial phases of research there. What I was addressing more was, well, what about when you show it sort of works in principle in a greenhouse or a big laboratory, and now you want to see if it works out in the field? How do you do that in that intermediate sort of scale-up research acceptably and safely? And that, I think, is where it's much more challenging and very hard to not do it in the environment itself. And where we might want to face is this a fine thing to do on a limited basis from small-scale research, understanding there could be some spread we can't contain and accept that as part of the conditions of the work. That's sort of what I was getting at in terms of, focusing on the product and its benefits and the risks thereof, rather than the notion that all gene drives, no matter what, must be fully contained until finally approved. So I hope that helps a little bit.
It does. You're really throwing a lot of emphasis on the risk-benefit conversation.
Yeah.
Yeah, okay. Other questions from committee members? Well, let me go again then. So, Steve, since I've got your attention, can you talk a little bit about what you would think as a funding model for these kinds of systems where there is a desire to manipulate organisms that it takes a while for these traits to be expressed? What are you thinking in terms of getting past the 3 or 5 year research award?
Yeah, that's been something I've been dealing with my whole career, and I don't have any easy answer. One thing that was very helpful for me and would apply here was the USDA-style CAP grants and the larger sort of NSF genomics grants that have more of a 5-year type of window possible. In fact, the field trial I showed you the images of with the RNAi trees was something that we put in because we were part of a USDA bioenergy CAP grant that had a 5-year duration. Otherwise, it's hard, and that's not really enough time for the establishment, analysis, and then getting rid of the trial. But it was enough to make it worthwhile to take the further risk. So I also feel in general, if we want, related to that issue, if we want applied translational biotechnology from crops and plants, that's not just coming from a few very large corporations, we want the public sector to have the ability to do that. There almost needs to be a translational biotechnology fund that allows that kind of thing. The USDA Biotechnology Risk Assessments Program now has a 4-year duration, but very, very small grants given the cost of this kind of thing. So I do think if we're going to do these kinds of things and do contained and semi-contained experiments with gene drives and GMO types of technology in general, we sort of really do have a desert in most cases, except for these very large programs that are exceedingly hard to get. So I do think a different funding model is needed if indeed we really want small companies in the public sector to be in the space.
If I could follow up real quickly, do you know of work being done, for example, within USDA or even USDA Forest Service, where they would have access to programmatic funding that could go on for decades?
I think there's a little bit in the U.S. Forest Service. For example, on the emerald ash borer problem I talked about, they've contributed to chestnut blight research for many years, continue to do that. It's very small and it's very, very spotty, but something does exist. And, you know, the political climate in the U.S. Forest Service and similar agencies for anything that's sort of biotech, meaning recombinant DNA, GMO technology, is very, very chilly and hostile. And so it's sort of a small miracle those programs exist, and so I'm glad they are. But I don't think there's anything on the scale we really need to face these kinds of forest stresses that I was talking about. And so I think we could quadruple it easily. And one of the practical problems is I think a lot of Forest Service money is consumed fighting fires, and that's a problem in terms of them being able to innovate more in this area and also many others. So the USA ARS, I think, has some programs like this. They certainly have a big program for the citrus greening problem. I don't know if it involves gene drive or not, but I think that's also a relatively rare case where biotech approaches are being used in an aggressive way, and I think that's mainly due to the chilly climate in USDA ARS.
I see. Thank you.
Yeah.
Questions, comments from committee members? Okay. Hearing none then, and we're close to the end of this webinar anyway, with only about 5 minutes to go. Let's bring it to a close, and let me thank our speakers, Dr. Mark Benedict and Dr. Steve Strauss, for really awfully interesting presentations, and presentations that I know are going to prove helpful to us as we prepare this report.